Hi, and welcome to another episode of Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Now today, we're going to talk about the origins of eukaryotic cells. It's most non-Darwinian. I know, it's weird. It's actually really cool. But before we get into the nuts and bolts of the origins of eukaryotic cells, you need to know first that it took several evolutionary innovations to kind of pave the way. One was photosynthesis, then the evolution of aerobic respiration, and then endosymbiosis. So let's go into each one of these key evolutionary innovations, and then I'm going to finish off with busting some of the popular myths about the way endosymbiosis actually occurred. I've made several videos on this already, but hey, I'll do it again. So oxygenic photosynthesis. Bacteria did it, right? It's like the Simpsons did it. Bacteria did it. No plant ever evolved photosynthesis, but bacteria did. Now, we all know that photosynthesis, photo means light, synthesis means putting together. The photosynthesis takes carbon dioxide and water and the energy in sunlight and puts them together to make carbohydrates. Now, a byproduct of photosynthesis, or at least oxygenic photosynthesis is, you already guessed it, it's oxygen, or free oxygen, O2. Now, free oxygen, most of us, we're like, oh man, we need oxygen. It helps us breathe. And we think that, life needs it. But the reality is oxygen is really detrimental to a lot of life. It's really reactive. So it easily damages organic molecules. However, because oxygen is reactive, it's what we call oxidative, life learned how to use that. So oxygen energized life. And over billions of years, oxygen levels slowly rose in the Earth's atmosphere, thank you to the activity of cyanobacteria. Now, like I said, oxygen's reactive stuff. It's bad stuff for a lot of life. So life evolved aerobic respiration. We typically just call that cellular respiration. And this was another key evolutionary innovation, paving the way for eukaryotes. So now we've got oxygen in the atmosphere. Now with aerobic respiration, we have an ability to use that oxygen and make a lot more ATP. You see, life needs energy, right? And the more energy you have, the more you can do with it. So life evolved the ability to use oxygen to make almost 3 times as much ATP as it could without oxygen. So now, got everything in place, right? We've got oxygen in the atmosphere, and we've got these aerobically respiring bacteria. And now for the third step, endosymbiosis. Now, endo means within, and symbio means living together. So endosymbiosis occurred almost 2 billion years ago when an aerobically respiring bacterium merged with an archaean, and then it went on to evolve into eukaryotic cells. And like I said, this is one of the most non-Darwinian evolutionary processes ever, right? Because Darwin is slow change over time. This is 2 cells merging and then evolving into eukaryotic cells. So endosymbiosis, this was incredible, right? I mean, for the first 2 billion years of life, there were only prokaryotes on this planet. There were no plants, no animals, no eukaryotes at all. It was all prokaryotes, right? So this sparked the largest cellular restructuring in 2 billion years. That's a long time, right? Now, if you remember, prokaryotes are limited in size, right, by energy production. So as these cells grew larger, they just packed in more and more mitochondria, right? And that would make more and more ATP. So eukaryotic cells could evolve to become larger and more complex compared to the prokaryotic cells, which remain mostly structurally simple. And then once eukaryotes evolved, it would be another billion years, and they would eventually go on to form plants, animals, and fungus. And in fact, all multicellular life today is made up of eukaryotic cells. There are no multicellular prokaryotes. There's none. It's pretty wild, isn't it? Now, this point that I'm making about prokaryotes remaining smaller and structurally simple is important because prokaryotes make the majority of their ATP across their cellular membranes. So as they get larger and larger and larger, we know the story where we're going with this, right? your surface area increases more slowly than your volume. Think about it this way. If you've got 1 by 1 centimeters, you have square centimeters. If you've got 1 by 1 by 1, you have 1 cubic centimeter. But if you have 2 by 2 centimeters, you've got 4 square centimeters of area. But if you go 2 by 2 by 2 in volume, you now have 8 cubic centimeters. And we go to the next one. If you have a 3 by 3, you have 9 square centimeters. And if you do 3 by 3 by 3 for volume, you have 27 cubic centimeters. So you can see that the volume increases much more rapidly than area. So if prokaryotes become larger, well, they actually run out of energy. And that's important. That's pretty cool. That's an important point to remember, because now I'm going to talk a little bit more about the origins of eukaryotes through endosymbiosis and how we know it didn't happen. I know in science it's nice to know how things happen rather than how it didn't happen. But let's continue on. So the origin of eukaryotes, this endosymbiotic theory was proposed in the 1960s by Lynn Margulis. And at first, she was not taken seriously at all. But by the 1980s, it became apparent that mitochondria were once free-living bacteria. They've got circular DNA, much like a bacteria. They have ribosomes that are more similar to a bacterial type of ribosome than a eukaryotic ribosomes. Now, they're both pretty similar in their function, but there are some differences we're not going to get into here. And guess what? Mitochondria can actually reproduce inside your cells by binary fission. So we can actually see how mitochondria were once free-living bacteria. And then we also know that the host cell was probably an archaean, and we base that on the fact of the way our DNA replicates. It replicates more closely to the way an archaean does it than the way a bacteria does. So that's really wild, right? Two prokaryotes, an archaean and a bacteria, merged to go form these eukaryotic cells. So let me talk about some common misconceptions here. The way it's written in most books, including the textbook I use, and the way we often see it on the web, is almost always wrong. They typically show one cell, one larger cell, engulfing a smaller cell. There's two problems. There's a larger cell, I talked about this, right? And another cell engulfing another cell. No. So I think these illustrations are wrong, and I think the textbook is wrong, and I'm not alone. I think a lot of cellular biologists agree with me on this one. For some reason, our textbooks, they get a little behind sometimes and need to be updated. So how do we know that an illustration like this one, it's a very nice looking illustration, and it shows this proto-eukaryotic cell getting larger with some membrane structures in there, some organelles, and it finally engulfs this thing that becomes a mitochondria. How do we know this didn't happen? Well, let's go with what we know. First, energy production. Remember, prokaryotes make ATP across their membranes. So as you become larger, your volume grows faster than your surface area, you run out of ATP. So this cell would likely not get that large because it would be limited in energy production. And even today, we don't see cells that are intermediates between prokaryotes and eukaryotes. We see either prokaryote or eukaryotic cells. Now, that being said, there are actually quite large prokaryotes, but the reason why they can do that is because they have invaginations in their membrane, giving them more surface area to make ATP. Now, secondly, prokaryotes don't do phagocytosis. Now, phagocytosis, this is where one cell can engulf a large object like another cell. In fact, our immune system does it all the time. We have white blood cells that will engulf cancer cells, bacteria. Now, why do we think that bacteria can't do this? First of all, we've never seen it in bacteria or any prokaryotic cell. We've never seen a prokaryotic cell engulfing another cell. And the reason why they can't do it, and likely never could do it, is because it requires a cytoskeleton of microtubules and actin filaments with motor proteins to move your membrane around another object and then pinch it off and then digest it with a lysosome. Bacteria don't have that. And secondly, this is like a highly energetic process. Now, as eukaryotic cells grow larger and larger and larger, you can have thousands, tens of thousands of mitochondria in them, making the ATP necessary for a very energetic process like phagocytosis. So, not enough energy for it. They lack the cytoskeleton elements to do it. And then third, most prokaryotes have a cell wall. And if you get a cell wall, your membrane's just pushing up against it. You don't move a membrane around another object if you got a wall around yourself. So, like I said, I'm not sure why most illustrations and most textbooks on the web don't get this right. All it takes is just some simple biology knowledge about our what limits a size of a cell, energy production, and the fact that prokaryotes don't do phagocytosis. They can't engulf another cell. Yeah, so I disproved stuff, but how did it happen? Good question. We really don't know. It's kind of a mystery. But that's the beauty of science. You see, we don't have all the answers, but that doesn't mean we stop looking for those answers. So what we think happened is that the aerobically respiring bacteria was likely an intracellular parasite. Intra, that means within. We do see bacteria that are capable of boring into other bacteria or other prokaryotes, and eukaryotes for that matter, and they can reside in there. So that's what we think happened, is that this intracellular parasite bored its way in there, and it started out as a parasitic or a plus-minus relationship. Then over time, that relationship became more mutualistic. The mitochondria, or the bacteria that became the mitochondria, became more and more dependent on its host, right? And it did so as it lost a lot of its genes. Those genes, they didn't just go away. They actually migrated to the nuclear genome. And eventually, these aerobically respiring bacteria evolved into what we consider the mitochondria, which are now completely dependent on their host. And the whole system, the archaea and the bacteria, are now what we call a eukaryotic cell. So that to me is just amazing, right? That we're a chimera. We're made up of two cells that merged. Like I said, very non-Darwinian, because the illustration I showed earlier of a cell getting larger and larger and larger and engulfing another cell is Darwinian, whereas the origin of eukaryotes, at least the kickstart to it, was not Darwinian. How cool is that? Okay, well, this has been another episode of Tom Kennedy's Science, and I hope you learned a little bit about endosymbiosis, because I think it's absolutely fascinating stuff. Until next time.